So here's your reward. Okay, so we're going to do blood today. We'll probably almost finish blood today. We'll do respiratory system after Thanksgiving. I'm almost finished with your test. Expect them to either be posted Wednesday night, tomorrow night, Thursday afternoon if I get really tired of my family and just want to hide in a room and do something that's not related to them. Or latest would be Friday morning because I'm giving myself Thursday if I need it. Okay, I'm almost done with them now. I just have to do the short answer part. I've done all the quiz recording and all the Scantron recording, all that kind of stuff. Okay, so it seems like there's something else I'm supposed to tell you. I don't know. So we are going to do blood today. So let me just give you an overview of what I'm going to do with blood. Primarily today, we're going to focus on red blood cell activity and then platelets. White blood cells are really freaking important, but I know that you get a ton of immunology and microbiology, and you don't get this other stuff in any other class. So we are going to talk about red blood cell synthesis and activity, and we're going to talk about clotting. Let's just acknowledge the overview of blood that you know, but may not have thought about in a little while. Basically, blood is plasma. Plasma is around 90% water with some clotting proteins in it and some other proteins, including albumin. And albumin is a protein that is primarily responsible for maintaining your blood volume. It's got so much, blood plasma has so much albumin in it that the albumin in the blood plasma sort of exerts an osmotic pressure to keep the fluid in. Does that make sense to you guys? Okay. It also has a little bit of pH buffering activity, albumin does. And also albumin, remember me telling you that for lipid-soluble stuff, getting them from point A to point B becomes problematic and sometimes they need an escort protein? Albumin is one of the really common escort proteins. So albumin, and then there's a whole bunch of clotting proteins, which we'll talk about today in the blood plasma. Anyway, blood plasma is mostly water, so anything water soluble is easy to carry in blood plasma. Ions, hormones, anything is easy to carry in blood plasma. Okay, and then of course we have our white blood cells. You know there's 5 different categories of white blood cells. We don't want to talk about them right now. It's a fascinating story. You can do an entire semester on immunology very easily. And then, of course, you've got your red blood cells. Remember that you should have learned before that red blood cells, mature red blood cells, don't have a nucleus or really hardly any organelles. Primarily, they're just little gas fairies. They carry gases, not flap flap, but toot toot fairies, right? Like a boat fairy. So that primarily they carry gases around, okay? And then platelets, do you guys remember that platelets are not whole cells anymore? They're cell fragments. Primarily, they're just a cell membrane bag filled with clotting proteins and a couple of other things. They can't go through cell division or anything, and primarily what they do is initiate clotting. Okay, so we are going to today skip down to Roman numeral 2 and just look at some general information about blood, and then we'll get into red blood cells. Okay, so general information about blood. Basically, blood is like take interstitial fluid, stuff it full of clotting proteins, and add a whole bunch of cells to it, right? Because your interstitial fluid, by the way, came from your blood plasma. It got pushed out into the tissue via filtration, right? So it's the same kind of stuff. So blood plasma has like the same ion composition as any other extracellular fluid, just with more proteins and stuff in it. So it is thicker than water. It's about 5 times as viscous as water. So it's about the viscosity of like whole milk. Although, of course, that can change depending on whether you're really hydrated or dehydrated. The viscosity can change. It is sticky because it's got sugars and proteins in it, so it tends to get kind of sticky. If you've ever had any decent amount of blood in your hands, it's like pretty sticky. And it's a little hotter than your average body temperature because it's going through highly metabolizing organs, right? And at any given moment, unless you're really hot, most of your blood is more toward the core than it is toward the periphery, which is a little warmer. And notice how tightly constrained its pH is. The pH is really tightly constrained with blood, 7.35 to 7.45. Does anybody know the 2 body systems that are really intimately involved in regulating your blood pH? What? Liver, it does a little, but not too much. kidneys, and respiratory, yes, because carbon dioxide, pH, that relationship, right? So in the next two sets of notes, we will do kidney regulation and respiratory regulation of blood pH. Your blood is isotonic to about 0.9% saline, right? That's isotonic. It makes up about 8% of your total body weight, and you usually have between 5 and 6 liters of blood, 4 to 5 in women, 5 to 6 in men. It's primarily just based on body size, not on specific sex. Okay, so let's talk about basic functions of blood. Well, you guys probably know what blood does. In part, it's transporting things from point A to point B, right, wherever you made it, like an endocrine organ to the target cell. Wherever you absorb oxygen at the lungs to wherever you need it, right? Wherever you generate carbon dioxide at the cells to wherever you can get rid of it, right? Taking things to wherever you can get rid of waste products. So it does an awful lot of transport. It also does regulation of your body pH because it's got pH, buffers in it. It does regulation of your ion composition from wherever you have a whole bunch of an ion to wherever you need it. So for instance, your bones have tons of calcium, but your brain needs calcium. So the fact that you have a circulating pool allows you to basically do that calcium conditioning, to take it from one place to the other. Your blood actually regulates its own volume because of the albumin that's within it, and also the capacity of the blood to clot, right? So it can keep from losing its own volume. So your blood protects you from microbes, toxins, pathogens because of the white blood cells and antibodies in it. Also protects you from blood loss because of clotting. And then your blood stabilizes your body temperature. And it does it by, again, that air conditioning effect from wherever it's hot to wherever it's cool, from wherever it's cool to wherever it's hot. And then also, since blood plasma is mostly water, you can absorb and release heat by breaking and forming hydrogen bonds, right? So, okay. So now we are going to get into red blood cell physiology. Ready? Okay, so you guys know where hematopoiesis, which is blood cell production, occurs. Where does it occur? Red bone marrow. Okay, now a specific subset of hematopoiesis is erythropoiesis, and erythropoiesis is specifically making red blood cells. That makes sense? And of course, that is going to occur primarily in your red bone marrow, yeah? And the process is called erythropoiesis. That good? Erythropoiesis. And so these are going to make your red blood cells and dump them into your bloodstream. But in order to make red blood cells, which of course are primarily about gas transport, you are going to need some nutrients and also some hormones in order to do that. So let's figure out what you need in order to make red blood cells. In order to do erythropoiesis, you have to have iron, right? Because iron is, of course, a major component of what? Iron is a major component of what? Hemoglobin, exactly. So hemoglobin is, of course, a protein that would be made in your red bone marrow during the process of erythropoiesis. You guys hopefully have seen hemoglobin before. It is actually a protein with a quaternary structure because it's got several different chains of amino acids, right? And it's got 2 alpha and 2 beta subunits. The alpha subunits are from one gene. The beta subunits are from another gene. 4 of them together makes a normal hemoglobin molecule. At the center of each glob of protein, globin, yes? As the center of each globin is a heme group. And at the center of each heme group is an iron atom, okay? And when oxygen binds to that iron atom, what happens is it rusts. And it makes it red, just like when iron actually is exposed to oxygen in the air. What happens? It rusts, it gets red, right? So when iron binds to the heme, it actually makes it bright red, right? And that is called oxyhemoglobin. And the blood, when it's got oxyhemoglobin in it, is bright red. When it's got deoxyhemoglobin, it's dull red, right? Okay, so you know this, but this is... Hemoglobin is actually an oxygen ferry, right? And it's got 4 seats for oxygen, right? Because the oxygen actually sits on the iron portion of the heme group, right? So you can carry 4 oxygens at a time. Okay, so let's talk about iron. So how do you get iron? How can you lose iron, right? Iron in your diet comes primarily from meat, from liver, from shellfish, from egg yolks, beans, nuts, cereals, leafy green vegetables. So if you ever have excess iron, it gets stored in your liver so that if you don't have enough, it can be released into your bloodstream. That is why if you eat the liver of another organism, you eat high levels of iron, which is super grody. However, when I was a kid, I was very iron deficiency anemic. My mom would make me spinach, which I loved, still love, as a kid. And she would also, being in the South, she would fry up chicken livers for me, yes, cut them into little cubes and stick them in a tray on the bottom of the fridge, and I would just toddle on in, right? Get the little chicken liver cube and eat it, and I'm not anemic anymore. However, probably you had really high cholesterol for a kid, because that's also where you store cholesterol. So that liver is actually the place where you store your excess iron if you ever have enough, if you ever have too much. So how do you lose it and how do you absorb it? The GI tract, your gastrointestinal tract, will control your rate of absorption of iron based on your body's needs. So generally you won't absorb more than you need if you are otherwise healthy. You'll just pee out the rest. And how do you lose iron? You lose iron because most forms of iron are water soluble. You lose it whenever you lose water. So you lose it via your urine, via your sweat, only a little bit, but a little bit. And then women lose a lot of iron via their menstrual flow, because when your endometrium sloughs off, you lose really vascular tissue. FYI, when you menstruate, you don't menstruate blood, you menstruate vascular tissue, right? You're actually losing tissue with a whole bunch of blood in it, right? So you end up losing iron during that process. And some women, it's not at all uncommon for women who are in their reproductive years and are menstruating to actually become slightly iron deficient and a little anemic during and right after menstruation, and then your level pops back up, and then you become slightly iron deficient, your level pops back up. That's not very common with men. Men's iron level tends to stay relatively constant in a healthy male over time. Okay, so you need iron. In addition, you also need some things in order to do cell division. Erythropoiesis is a big cell division party, right? So you have to basically do cell division and cell division and cell division. As a precursor to cell division, you always have to do DNA synthesis before cell division, right? Double it before you split it. Double it before you split it, right? So in order to do cell division and DNA synthesis before it, you need folic acid and you need vitamin B12. Not just for red blood cells, but for cell division anywhere, you need those two things, okay? Folic acid, also called folate, is necessary to basically, it's one of the constituents for making thymine, which is the ATGC, it's the T. So sources of dietary folic acid, green plants, yeast, liver, and citrus are the primary places that you would get that. You need adequate amounts of that in order to do red blood cell synthesis. And then vitamin B12 is a biggie. The dietary sources of vitamin B12, I say only found in animal products, but it's really only found easily accessible and in high levels in animal products. This is, of course, a big deal for vegans, right? If you are a vegan, you need to make sure that you are getting high enough levels of vitamin B12 in order to do cell division. And there are ways to do it. It's just a little harder when you're not eating animal products. Okay, so I want to take just a second and talk about folic acid, even though it's not directly related to red blood cell synthesis. I feel like I would be remiss if I didn't talk about this. Folic acid is absolutely necessary for cell division, for the DNA synthesis precursor to cell division. Pregnancy is a big cell division party, right? Okay, now, one of the things that folic acid we know is absolutely necessary for is the development of your nervous system, the development of a fetal embryonic nervous system. And it's necessary during the entire pregnancy. So one of the things that they started doing about 10 or 15 years ago is they figured out that some women were folic acid deficient, and that was really easily remedied. And also, if they didn't remedy it, it would cause serious nervous system development problems like hydrocephalus and spina bifida, because it's needed during even the first 2 weeks of pregnancy as your nervous system, the fetal nervous system starts to develop and sort of put down the framework for where it's going to be. Your nervous system starts out as a flat sheet, and then it starts to fold, and it folds anterior to posterior. If you don't have adequate levels of folic acid, it's going to fold, and your ventricles will form abnormally. right? You can get hydrocephalus associated with that. Or your neural tube will not close all the way down at the bottom, and that's called spina bifida. Those can cause paralysis and permanent brain damage. So about 15 years ago, they said, well, we can actually help women prevent an awful lot of birth defects by letting them know that they should be taking folic acid. So who should be taking folic acid? Every single woman who would not terminate a pregnancy if one would occur. Because when do you need folic acid? First 2 weeks of pregnancy. How many of us who are moms knew we were pregnant during the first 2 weeks of pregnancy? No, we didn't know we were pregnant, right? We were still like, life is good. And you're like, what the? You need it the whole time. And so if you're not going to eat really well and you wouldn't terminate a pregnancy, you actually need to make sure that you are taking a multivitamin with folic acid. And you can prevent those neural tube defects. How many chances do you get to put together a nervous system? One shot. That's all you get. There's a song about that. One shot. Do not miss a chance. Okay. My little boy is obsessed with Eminem. He's 11. I know it's inappropriate. It's still hilarious. I did get to teach him about homophobia and racism because we were listening to Eminem, though. I was like, I'm going to teach you a word. It's called homophobia. And even though it's got a good beat, the message is a little dodgy. Okay. So erythropoietin is next. This is a hormone. I know you want another hormone like you want a hole in the head. This one's kind of cool. Okay. Erythropoietin is made by the kidneys in response to hypoxia, low levels of oxygen. And what it will do is long-term take care of that problem for you, but not short-term. Okay. So what it's going to do is when the kidneys detect low levels of oxygen over decently long periods of time, they're going to go, hey, we're still low in oxygen. Hey, we're still low in oxygen. Then the kidneys will release EPO. And EPO will go to a place that can help you take care of the low levels of oxygen being delivered to the kidneys, but only long term. Where will they go? Where will it go? Where's the target for EPO? Bone marrow, yeah. It's going to go to the bone marrow. It's going to force erythropoiesis. But is that a quick fix? No, it's not a quick fix for hypoxia. It's a long-term fix for hypoxia. Does that make sense? So it's going to go to the bone marrow and cause erythropoiesis. So it targets red bone marrow stem cells, and it causes erythropoiesis. So here's what it does. Kidneys release EPO, goes to the bone marrow, and causes erythropoiesis, assuming that you had all of that stuff that you needed for erythropoiesis, folic acid and B12 and iron and all that business. Okay, so erythropoiesis is bone marrow cell division. The body can produce 2 to 3 million red blood cells per second. And they only live about 120 days, so you're having a constant turnover of red blood cells. So let's talk about what could trigger EPO release. So we know it's hypoxia, but why could you be hypoxic, right? You could be hypoxic because you just gave blood or you just hemorrhaged. Does that make sense? If you just gave blood, wouldn't you be hypoxic? Right? You wouldn't be delivering adequate blood, oxygen to anywhere, including your kidneys, right? So immediately after you give blood, you're going to be hypoxic and you're going to generate EPO and then cause red blood cell synthesis. How long does it generally take after giving blood to replenish your blood volume? Usually about 2 to 3 weeks, okay? Do not do what I did when I was 18 and gave blood for the first time. That's the first time I could give blood when I was 18. And I was not the brightest bulb in the chandelier. Immediately after I gave blood, it was one of those days where I'm just going to do all of the things that make me feel good about my life. I gave blood, and then I went and worked out. It took me 72 hours to recover from that. I thought I was going to croak. Okay, that is not smart. In my defense, I was an English literature major at the time, so I didn't know a damn thing about a damn thing, right? Except Shakespeare. I knew some stuff about Shakespeare. Okay, so don't do that. Decreased availability of oxygen can also trigger hypoxia. How many people besides me moved here from low elevation? Yes. You move here and you get here and you're like, first of all, where are all the brown people, right? If I look exotic, you know you got some diversity problems, right? Like, just because I'm not blonde, you're like, oh my God, what are you? And I'm like, I don't know, Native American? Like, I was here first, honestly. So then you also figure out that you can't walk up the stairs, right? You're exhausted. And so what happens is your body gets an error signal about hypoxia, right? It's saying, you're not getting enough oxygen, you're not getting enough oxygen, you're not getting enough oxygen. My level of oxygen was fine for Texas because I was at sea level, right? And there's way more oxygen in the atmosphere, so I didn't need as many oxygen ferries. Yes? And then I moved here. I couldn't walk up the stairs without being winded. Gosh. And then my body was like, are you going to stay? Because it needed that error signal, not for a short period of time, but a consistent error signal for a decently long period of time. And then as soon as my body was sure that I was actually going to stay, then it starts releasing EPO, targeting my bone marrow to make more red blood cells, yes? Okay, make more red blood cells. How long does it generally take you to increase your red blood cell synthesis? Usually about 2 to 3 weeks, right? And so you couldn't walk up the stairs and you looked around, right? You're like, I don't even know why the hell I'm here. And then eventually you could walk up the stairs, right? You're still in Utah, but it's a nice place, honestly, all things considered. So you do erythropoiesis when you get to high altitude if you've been at low altitude. COPD is chronic obstructive pulmonary disease. This is a disorder that causes, it's a really big umbrella disorder that causes problems with getting adequate levels of oxygen into your system. It could be triggered by chronic bronchitis, by pneumonia, by almost anything respiratory. But can you see how COPD would also cause erythropoiesis? Yeah? Okay. And then increased demand. If you start exercising, then your skeletal muscles are going to gobble up more oxygen, and so your kidney is going to get less, right? Now, exercising once, does that do it? January 2nd, January 3rd if you're hungover, right? That's what everybody does. You know they sell all of those memberships, right? In January, you're like, this is the year that I'm going to get hot. Let's just take a deep breath and say, this is probably the hottest you're ever going to be at this particular point right now, okay? I mean, how often does it change, really, for the better? Not so much. But you might be able to afford nicer clothes, right? Good tailoring, it'll be fine. Increased demand, exercise will cause it. One last little tidbit about decrease in red blood cells because of hemorrhage or destruction. Malaria is an internal red blood cell parasite that causes hemolysis. Does it make sense to you that someone infected with malaria would therefore have high levels of EPO being released? Does that make sense? So hold on to that because we'll come back to it in a minute. Okay, so EPO causes erythropoiesis. It has now been genetically engineered so we can make it in the lab. Where did we used to get it? This is not a happy story. I feel absolutely terrible when I look this up. So what we used to do before we figured out how to genetically engineer erythropoietin to make it in the lab is we used to buy the urine from people who were infected with malaria. I'm really hoping that we also gave them medication, but I don't really trust us, do you? No, I don't trust us. So we used to buy the urine because they made so much EPO when they were infected with malaria that they ended up peeing out a ton of it. And then we would extract the EPO from the urine of people infected with malaria and then use it to treat our patients. Now we don't do that anymore. Now we actually make it in the lab, and now it's more readily available. It's a little cheaper, right? How is it used clinically? And then we'll talk about how it's used off-label. How it's used clinically is if somebody had leukemia, one of the treatments would be we needed to blast their bone marrow, right, to get rid of the cancer. But then the problem is when you blast their bone marrow, in addition to other things, you have too few red blood cells to do adequate healing, right? So what if you could shoot them up with EPO to get more red blood cell synthesis, right? That drug is called Procrit. That's one of the brand names for that drug, and it's used for a bunch of different treatments. Now, of course, EPO is more readily available. So what are some off-label uses for EPO? Anybody heard? Yeah, blood doping. So if I were a competitive cardiovascular athlete and I wanted an advantage over the other competitive cardiovascular athletes, increased oxygen carrying capacity would do it for me, wouldn't it? Yeah? So I could shoot myself up with EPO and increase my red blood cells, right? And then get a competitive advantage. Or there's another way to do blood doping that isn't directly injecting you with EPO, but it's forcing you to make it. And that is that I can give my own blood, right, to myself. They spin it down, they pack the red blood cells, you keep it in the fridge for a few weeks with some anticoagulants to make sure it doesn't clot, right? And then you allow yourself 2 or 3 weeks to regenerate your red blood cells, and then you shoot yourself back up with your own red blood cells. That's another form of blood doping. Either one is actually illegal in competitive sports, but very difficult to detect. First of all, the EPO that they produce in the lab is damn near identical to the EPO that you produce, right? So detecting that level doesn't do a hell of a lot for you. And then second, competitive cardiovascular athletes have high red blood cell counts anyway. So what they finally decided to do, which sport is famous for blood doping? Cycling, yeah. And Lance Armstrong admitted that he did blood doping. So now what they did, at least the last time I checked, if somebody knows something different, tell me. Now, the last time I checked, what they said is, I don't care how you got your red blood cells, if you have more than this level, you cannot compete, right? Doesn't even matter how you got them. So you better just sit on the couch for 2 weeks if they're too high. So, and the level, by the way, is not a level that any of us would get to, right? It's excessively high levels. It's like really competitive cardiovascular athletes. Okay, so the other things that can cause, oh, by the way, is there any potential negative to having too many red blood cells? What is it?
High blood volume.
High blood volume? What else?
I was actually going to ask you about that. What would lead to excessive high levels of blood?
Well, if you were doing it naturally, your homeostasis will not let your levels of red blood cells get so high that it becomes problematic. But if you are doing blood doping, any problem with shoving more and more solids into your bloodstream? What can it do to the viscosity of your blood? It's called erythrocytosis, and it can be like pumping maple syrup, and you can have pulmonary embolisms, heart attacks, and strokes associated with erythrocytosis. So it is dangerous, and competitive athletes have died because of erythrocytosis because of blood doping. But if you're naturally doing it, your body will just say, that's enough, don't make any more, right? Okay, so what are normal levels of red blood cells per cubic millimeter, which is a milliliter, big drop, right? Usually males have about 5.5 million, 5.4 million per cubic milliliter, and females have almost 1 million less per cubic milliliter. And that is because testosterone causes erythropoiesis. Testosterone actually causes you to release EPO. So males have greater oxygen carrying capacity than females have, because of course they've got testicular testosterone in addition to adrenal cortex testosterone, right? So if you were competing against a cardiovascular athlete, even at the elite levels, do the male and female cardiovascular athletes compete against one another? They generally don't. Does that mean that a female could not beat a male? Of course she could, right? Serena Williams could probably kill anybody else on the tennis court right now. And of course, Billie Jean King did beat, what's his name? I can't remember. Sexist male tennis player said that any elite male could beat any elite female, and she whooped him in a big, broadly publicized. But if we're talking about populations, right, males have greater oxygen carrying capacity. In addition, they've got more muscle mass, right? So you can whoop them at chess, okay? They might beat you at tennis, fine. Unless it's Serena, and then she's going to beat all of us at tennis, isn't she? Right? Okay, so males have greater quantities of EPO, therefore they have greater oxygen carrying capacity, and they're going to have more hemoglobin as well. Okay, so now let's talk about hemoglobin just a little bit. So hemoglobin we already talked about, but of course it is a red iron-containing pigment. This is the heme portion. This is the globin portion, yes? So it's hemoglobin. Globin was actually named because it's a globby protein, so they named it globin, yes. And of course it makes blood red. Now, since males have greater numbers of red blood cells, they have greater hemoglobin count, right? So if you dried down the hemoglobin out of about 100 mils, males would have about 13 to 18 grams and females would have about 12 to 16 grams, okay? And there's variations, right? Athletic activity actually makes you release more testosterone, by the way, so female athletes would have more than other females, right? They actually release more testosterone. And did you know that winning things actually makes you release testosterone too? Like as soon as you win something, you get a little squirt of testosterone, which makes you want to win more stuff, which is interesting. Okay, so functions of red blood cells, you already know this, but red blood cells' primary job is gas transport, but it doesn't just carry oxygen, it also carries a little bit of carbon dioxide. So let's look back at hemoglobin for just a second. When oxygen sits on the seats in hemoglobin, there are 4 seats for it, 1, 2, 3, 4. When oxygen binds to hemoglobin, it makes it redder. It's called oxyhemoglobin, okay? And almost all oxygen is carried this way. And so what you would do is you would form oxyhemoglobin in the pulmonary capillaries, right? Form it. Oxygen load up onto hemoglobin. Then you would go to the heart, and then you would pump it to systemic capillaries, and then you would unload it off the ferry, right? But at the same time, you were unloading the oxygen off the ferry into systemic capillaries to be used for cellular respiration. You can also pick up carbon dioxide, but carbon dioxide doesn't sit on the same seats. So it never outcompetes oxygen because it actually sits on the globin portion and not the heme portion, right? Does that make sense? So I don't know if you guys know this, but knew this before, but hemoglobin also carries carbon dioxide. It only carries about 20% of the carbon dioxide. The rest of it is carried differently. But anyway, it does both of those. Okay, so let's talk about what happens to your red blood cells as they move through your system. As they move through your system, they last about 120 days, but they get the crap beat out of them as they move. Because some capillaries are barely bigger than a red blood cell, and some of them have really acute angles in them. So as they're sort of folding around the angles, they just kind of get destroyed eventually. And so you're constantly looking at your red blood cells to see if they've got another round left in them. And the primary place that you do destruction of red blood cells when they're worn out is the spleen and the liver. And the spleen and the liver both have big sinusoid capillaries. Do you remember sinusoid capillaries? Sinusoid capillaries are these big, wide capillaries, right, with big holes in them. And so I want you to imagine that red blood cells are going through the spleen and the liver, and there's this big, wide capillary, so it's slow. And it's got a big hole in the top and a big macrophage called a Kupffer cell sitting on top of the hole. And so as the red blood cell goes by, if it looks funky, and you're like, you've had it, bro, it like reaches down and does phagocytosis and gobbles up everything useful, dumps the good stuff back into the bloodstream for use, and then trashes the bad stuff. So what kind of good stuff does it gobble up and then dump back into the bloodstream? Iron, right? Sends it right back to the bone marrow so you can make more. The amino acids from the globin, you dump those into the bloodstream and you can use them for whatever the hell you want, right? Anabolic or catabolic processes. And then you have these waste products that you can't actually get rid of, and those are eliminated as bilirubin, which is this super grody green color, which makes it into your bile. And then bile gets dumped into your digestive tract primarily, although a little bit of it ends up in your urinary tract. And bile has some good functions, but the bilirubin in bile doesn't really do anything except make your poop that unpleasant color that you would never like decide to paint a wall. Except maybe this wall, what the hell? That's like not, that's almost poop, isn't it? Like greeny brown, right? Who thought that was a good idea? So yeah, it makes your poop that color, and it also makes your pee that color. And don't think that 11-year-olds do not love the fact that it's like broken down red blood cells that make their poop that color. Unless, of course, you've taken vitamins, and then your pee is that color because of vitamins. Okay, so let's talk about clinical connections. Anemia. You guys, I know you've heard the word anemia, but anemia is broader than it's usually used. All anemia means is reduced oxygen carrying capacity, and it could be because of weirdly shaped hemoglobin, too few red blood cells, normal numbers of red blood cells, but not enough hemoglobin in the red blood cells, any number of things. Regardless of how you get anemia, does it make sense to you that it tends to be associated with pale skin and kind of fatigue and exhaustion, both mental and physical, right? And a lot of times you're a little cold. So let's look at some examples of anemias. What kinds of things can cause anemia? Hemorrhagic anemia means you just either gave or lost blood, right? So immediately after you give blood, you're going to be anemic, and you shouldn't go work out, idiot. So hemorrhagic anemia is anemia due to blood loss. That's going to go away, right, after you regenerate. You're assuming that you live through it. It's going to go away after you regenerate your blood. Hemolytic anemia is anemia because of destruction of your blood cells. Like if you had a mismatched blood transfusion, then the cells would agglutinate, and then it would cause hemolysis, and your cells would get destroyed, and then you would have hemolytic anemia. Or malaria causes hemolytic anemia. Isn't that the kind that it causes? Because malaria infects the red blood cells and then causes lysis to spread the pathogen, and then it infects the next red blood cells and causes lysis. So that's hemolytic anemia. Aplastic anemia is when the problem doesn't occur in the blood, the problem occurs here in the bone marrow. So if, for instance, I was at Chernobyl right after the explosion, the radiation that I encountered might destroy my bone marrow and cause aplastic anemia, which doesn't just cause red blood cell problems, anemia, also depresses your immune system because you lose your white blood cells, and causes problems with clotting because you lose your platelets, because all of those come from the bone marrow, right? Okay. Sometimes aplastic anemia is a side effect of chemotherapy or radiation treatment, right? Does that make sense? Because you have to kill it because it's actually trying to kill you, right? So that's aplastic anemia. Nutritional anemia is when you are missing one of the components that you need in order to do erythropoiesis. So a lack of vitamin B12, a lack of folic acid, a lack of iron, a lack of protein, those can all cause nutritional anemia. If you have a woman who is reproductive age and menstruating and also doesn't eat well, then you got a double whammy, right? You have a little bit of kind of hemorrhagic anemia, not exactly, but a little bit, right? And then you've also got nutritional anemia. So how do you take care of the anemia when you're during those years? Well, you make sure that you're eating well, right? When you know that you are going to menstruate, otherwise you are going to be super likely to have the anemia associated with menstruation. Does that make sense? Okay. Renal anemia, if something is wrong with your kidneys, they will not release EPO necessarily. So for instance, if you're on dialysis and your kidneys are completely failing, one of the things that they forget how to do is to release EPO, and so you will get renal anemia. Does that make sense? They don't release EPO. And then pernicious anemia, I love. Pernicious, doesn't that sound like old people? Pernicious? It is old people anemia. This old people anemia is because you can only absorb vitamin B12 when you make a factor called intrinsic factor and dump it into your gut. As you age, you make less and less intrinsic factor. So even if I ate vitamin B12, would I absorb it? No. And then I would get anemia and I'd be super exhausted because of that. Would vitamin B12 pills help these people? No. What do they have to do? They have to inject it. Right? They have to have vitamin B12 injections as they start losing that. Does that make sense? Okay, and then the last one, you guys have probably heard of this one before, but I just want to discuss it briefly, and then we will get into platelets and blood clotting. Okay, hereditary anemia is the hereditary anemia, sickle cell disease, which used to be called sickle cell anemia. Have you guys heard the sickle cell story before? Okay, I'm just going to do a quick version of the sickle cell story then. With sickle cell disease, what happens is it is a genetic disorder that causes an abnormal hemoglobin conformation. And this abnormal hemoglobin conformation, if this person on the left gets into a hypoxic situation, their cells will sickle and they'll look like the one on the right. Does that make sense? Okay. Now, if capillaries are barely big enough for red blood cells to go through at the best of times, how well does it look like a sickled red blood cell would go through? No, it causes a sickle cell crisis and it blocks up your blood cells, which can of course lead to a heart attack or a stroke or a pulmonary embolism. And even if it doesn't, it's incredibly painful, incredibly painful. Okay, so the irony is that it's also relatively common in people from certain portions of the world. So it affects a big swath of people from the African continent, or originally from the African continent. It also affects, there's a decent chunk from the Asian continent because it's got this link with malaria. So let's talk about the link with malaria. Okay, so first off, in order to have sickle cell disease, I have to be homozygous recessive for sickle cell. 2 different mutated genes for sickle cell would mean that whenever I got into hypoxic situations, my cells would sickle. And that's deadly. Okay, not immediately, but usually people with sickle cell disease will die before they reproduce. So usually adolescents, historically speaking. Now they're living longer now. There's some more treatments for it. So if you usually die before adolescence and you need a double dose of it, why the hell is it so common still, right? Why do you have 60 to 80,000 people have sickle cell disease? Do you anybody know anybody with sickle cell disease? I know somebody with sickle cell disease. Okay. So the reason it is so common is because although homozygous recessive has sickle cell disease, heterozygous is actually protected against malaria. Because malaria is an internal red blood cell parasite. And apparently if you're carrying one sickle cell gene, you don't sickle, except in extreme situations. You probably not even know that you were carrying it. You don't sickle, but the malaria pathogen will not actually infect your cells. So now we've got the heterozygotes that are more likely to survive malaria, right? And when you get two heterozygotes that breed, right? Their baby, what's the likelihood that their baby would be homozygous recessive? Two heterozygotes, but you do the Punnett square in your head. Two heterozygotes breeding, probability of a homozygous recessive is 1/4 every time. 1/4 every time. And so since heterozygotes have a distinct heterozygote advantage, they've survived in the population for a long period of time because of their resistance to malaria. Does that make sense? Okay, so that's hereditary anemia, sickle cell disease, and there is a link to malaria. There's also other anemias. If you never exercise, you don't need very many red blood cells, so you're a little bit anemic. Hypothyroidism. If you are hypothyroid, then the metabolism of your red blood cell production is also affected, and so you tend to have fewer of them. Go ahead.
So when you start exercising, how long does that take to start?
Usually about 2 to 3 weeks, if you are consistent about exercising. Yeah. Okay, so next thing that we are going to do is we are going to switch from red blood cells to platelets. This will seem really hard at first, but it will simplify if you stick with me, okay? So we are going to be talking about platelets and hemostasis. Hemo, not homeo, right? Hemostasis is the cessation of bleeding after an injury to a vessel, okay? Is it homeostatic? Well, hell yeah. But would you get credit if you put homeostasis when you meant hemostasis? No, that's not a spelling error. That's a different word, okay? So hemostasis is the cessation of bleeding when blood vessels are damaged. Before we get into the nitty-gritty of it, I want to talk about the big picture of hemostasis. So what I want you to imagine is that we have damaged a vessel, okay? It is going to be easier to stop the bleeding in which? An artery or a vein? Vein. Yeah, it's going to be easier to stop the bleeding in a vein. So let's say vein. We've damaged a vein, okay? And I want to know the stages that lead to the cessation of bleeding and ultimately the repair of the vessel. So I'm going to go through all 5 first, and then we'll go back and add detail. So you don't need to flip through with me the first time. So the first thing is when you damage a vein, you should do vasoconstriction, because when you damage a hose and you want to stop losing fluid, you kink the hose, right? So the first stage is kink the hose. Kink the hose upstream or downstream of the hole? Upstream, yes, of the hole. So that is called vasoconstriction, and that makes perfect sense. Vasoconstriction lasts for about 30 minutes because it takes a little while to fix the hose, right? Okay. The next stage is platelet plug formation, or as I call it, the platelet party. You got to start a platelet party because the platelets are going to gather together and they are going to sort of get in the way of the hole. It is not a clot. It's a platelet party. And what the platelets do is they basically start attracting their friends. And if you let them sit there unsupervised, the next stage will occur too. Somebody's going to get pregnant. No, you're going to form a clot is what's going to happen, right? So after you start your platelet party, if you let them congregate unsupervised, what will happen is they'll start the next phase. And the next phase is coagulation or clot formation. A platelet party is not a clot, but it leads to a clot, right? A room full of teenagers is not a pregnancy, but it leads to a pregnancy, okay? So here is a clot. A clot is when you start to make these insoluble, sticky, fibrous, spiderwebby things. It's made of a protein called fibrin, okay? And think of them as spider webs because what they do is they will stick to the hole and they will catch anything that goes by. Like those red blood cells, they don't have to be there, but they got caught as they went by, like a fly in a spider web. And then you have all of these things, and the little white one that you see in the middle there, that's a platelet. Those things, that sticky, gummy stuff, will actually help plug the hole. That's a clot. Okay? Does that make sense? Okay. And then the next phase, so we were in phase 3, so we've got vasoconstriction, platelet party, right? Coagulation, right? Next phase is clot retraction. And clot retraction happens when you pull on the spider web, and it pulls, because the spider web is actually stuck in the hole, it pulls the sides of the hole together to make it easier to heal. Does that make sense? That's called clot retraction. Have you ever had a road rash? And then after you have a road rash, you actually get to that period when it starts to pull together and it pushes out that clear yellow fluid? That's clot retraction. And what you're pushing out is serum. It's blood plasma minus all the clotting proteins that stay in the road rash, right? The scab is actually the clot, okay? And then the last phase is to dissolve the clot when everything is fixed, right? So 5 stages of hemostasis: vasoconstriction, platelet party, coagulation, clot retraction, dissolve. Good? Okay, so what we're going to do is we're going to go through each one of them. There's only one hard phase, and it seems hard at first, and then it gets simpler. Okay, so first phase, vasoconstriction. It is what you think it is, but it only works in vessels with smooth muscle. So what kinds of vessels cannot do this? What kind of vessel does not have smooth muscle in its wall? Capillaries, yes. So capillaries can't do vasoconstriction. They don't have any smooth muscle. Any other kind of vessel can do vasoconstriction because it does have smooth muscle. So the smooth muscle in the vessel wall constricts. It's basically like kinking the hose. The vessel gets smaller, reduces blood loss. The cause, I put a question there because we actually don't completely understand the cause. They think part of it is just a nervous system reaction to damage. You poke it and then, right? But the other is that you actually release paracrine agents that will cause the smooth muscle to vasoconstrict, right? And the paracrine agents are weird things that you wouldn't expect to have this function. However, you know if your body knows how to make something, it can use it any way it damn well pleases, right? So now we are going to use ADP, of all things, as a paracrine agent to cause vasoconstriction, and serotonin is a paracrine agent. And then another one called thromboxane A2. Anytime you see the prefix thrombo, it means what? Clot, yeah. So we're not at a clot yet, but we can predispose to a clot. Okay, so I did vasoconstriction. This lasts up to 30 minutes because it takes a little while to fix the hole. We have to spackle the hole over and then wait for it to heal. Okay, so the next phase is called platelet plug formation, or your platelet party, right? So what's going to happen is these platelets are going to start to congregate. So let me talk to you about something first. Okay, so do you remember a little bit about the structure of a vessel wall? So try to see if you can recall the structure of a vessel wall. First general question: the inner layer of a vessel that's close to the blood cells should be what general kind of tissue? You don't have to get the specifics right. Epithelial, connective, nervous, or muscular? Epithelial, yes? And does epithelial tissue have collagen in it? No, it does not. Okay, so does it make sense to you that the platelets would know that something was amiss if a platelet came into contact with a collagen fiber? Yes? That's a decent indication of damage, isn't it? Because the tissue that is immediately deep to the epithelial tissue in the basement membrane is loose areolar connective tissue with collagen fibers in it. So here's what happens. When a platelet actually comes into contact with exposed collagen, that's the trigger that says something is wrong here, right? And what that does is it changes the platelet just a little bit. It actually starts making it release some paracrine factors that will activate the platelet. What is an activated platelet? An activated platelet is a platelet that is slightly polar, meaning that it's got a positive end and a negative end. And if you've got a positive end and a negative end, and you actually call your friends in and they've got positive and negative ends, what happens is you start forming a little blob, right? A little kind of like polarity blob, right? And then as you start forming the blob, everybody sends out a group text and says, platelet party my house, right? And the group text is paracrine agents. More thromboxane A2, more ADP, more von Willebrand factor, all kinds of paracrine agents that start a little positive feedback loop, yeah? And the positive feedback loop is because you release all of these paracrine agents that start attracting more platelets and more platelets and more platelets. So epinephrine is released, serotonin, prostaglandins, ADP, thromboxane A2, and you get this platelet party, platelet plug. This is not a clot, but if you let it sit there for a second, it's going to form one, okay? It will start one. Okay, so this platelet mass grows in a positive feedback loop in which you just keep basically attracting more friends to the area. This usually takes about 1 minute, okay? It's relatively effective if your blood flow is not super high, but in arteries, right? Unless you kink the hell out of an artery, you're not going to be able to plug it, right? This platelet plug takes about a minute to form. It might be big enough to plug a teeny tiny hole, but most of the time it's not adequate and you need to go to the next stage, which is clot formation, okay? So, question for you. Why doesn't this thing just keep growing like a big behemoth and take over the entire vessel since a positive feedback loop? And the answer is that although the platelets are attracting other platelets and going, platelet party my house, platelet party my house, the healthy cells around are going, get off of my lawn, and releasing anti-platelet plug formation factors. And these are saying, go away, go away. So what happens is you're doing positive feedback at the site of damage and negative feedback on the sides, right? Does that make sense? The healthy, undamaged tissues are releasing things that say, move along, right? Nothing to see here. That makes sense? So what it does is it sort of hems in your platelet plug and keeps it just in that location where you had damage. Good? Okay, so basically, the platelet plug is not going to be the same thing as a clot, but will often precede a clot. And we will see that why it will precede a clot in just a second. Okay, so let's talk about aspirin therapy. People with cardiovascular abnormalities, predispositions to stroke, atrial fibrillation like I have, valve disorders, are often told to take daily aspirin therapy. And the daily aspirin therapy is because, do you guys remember that aspirin decreases prostaglandin secretion? And prostaglandin is one of these factors that causes clot formation. So I'm just taking out one of the factors in the positive feedback loop, right? And making it less likely that I will form inappropriate clots, right? Just by taking a daily 70 milligram aspirin or 80 milligram aspirin every day. Does that make sense? So what I'm doing, they call it an anticoagulant, but it really isn't. It's actually anti-platelet party, right? And then the platelet party would lead to a clot if it formed inappropriately. Does that make sense? So that's the daily aspirin therapy. Okay, so now we get to the hard part, but it will get as easy, I promise, if you just stick with it. Okay, so the next phase is the coagulation phase. And the objective of the coagulation phase is to make these sticky spider webby things that will, if you let them, plug the hole and spackle it so that healing can be occurring behind. Does that make sense? Right? So you're basically sort of plugging it. You're not going to leave it there forever, but it's plugging the hole so I'm not losing fluid as the healing is going on. Good? Okay. And the way that you do this is it's going to be more likely to start if you have platelets in the area. So we'll see that in just a second. Okay. So general information on clotting. If you've ever drawn blood or actually dropped blood on a table, you will see that it actually sort of forms a little bitty gel. It becomes a little bit more solid if you let it sit around. And the reason it becomes a gel is because it actually gets these fibrous proteins within it. And if you let it sit there for a minute, I don't know if you've ever done this, if you've ever dropped blood on a table or a slide and let it sit there for a second, as it becomes a gel, it'll actually squeeze out clear yellow fluid at the edge, right? And as it does that, basically all the clotting factors are in the inside and you're squeezing serum out on the outside. So drawn blood will thicken and it will gel. The gel is a clot made primarily of this protein called fibrin that's insoluble until you actually release an enzyme that dissolves it. And the way that you make this is by activating a cascade of inactive proteins that were always present in the bloodstream. So why do I not just make each protein when I have damage? Because it's going to be too slow, right? And we want clotting to occur quickly, correct? Okay. So you remember with the renin-angiotensin-aldosterone system that we have these inactive, right, proteins. we always had angiotensinogen and we always had ACE, right? But they didn't actually interact with one another unless they got that renin trigger. So the same thing is going to happen here. We have all of these inactive proteins in the blood plasma that could add to clotting, but they're never going to interact with one another unless somebody tells them that I need to plug a hole. Okay? Does that make sense? So it's going to be a cascade in which this one activates that one, and that one activates that one, and that one activates that one. Does that make sense? So it's a cascade and it's pretty fast. It looks super complicated, but I will be able to simplify it for you. So it's basically a domino effect, and it involves a whole bunch of clotting factors. Most of them come from the liver. So the liver is intimately related to blood clotting. So now before I show this to you, I want to tell you that all of the clotting factors are named by Roman numeral, but the Roman numerals don't have a damn thing to do with the order in which they're activated. They're the order in which they were found. So you have like 12 and then 8 and then like 3 over there. So don't worry about that too much, okay? And the other thing is that 12 and 12A are just 12 was inactive form and then 12A is activated form. So there's half as many as it looks like because A will always follow. Right? That makes sense? Okay, so I'm going to show you this, but I promise it'll get simpler in just a second. Okay, so here is the coagulation cascade. Okay, here is where we're trying to get. We're trying to get to fibrin because fibrin is the spiderwebby stuff, right? That's where we're trying to get. Okay, so the coagulation cascade, there's going to be 2 different ways to start it. You with me? Intrinsic pathway and extrinsic pathway. We'll come back to that in just a second. Regardless of which way you started it, you have to get here, which is factor 10A, which is an enzyme called prothrombinase. So what we're going to do is we're going to do this little part first, and then we'll come back and figure out how to get to 10A. You with me? Okay, so clotting will take usually 3 to 6 minutes, and it requires calcium at multiple points. So before we go any further, I've told you all semester long that clotting requires calcium. And now I want to look. Do you see calcium? Calcium, 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 calcium. So required at multiple points. Okay, so there's 3 stages in the formation of a clot. The first one is getting here, factor 10A. I'm going to come back and pick that one up, okay? And then after you get 10A, you're going to actually do this stuff. So the first one is the production of prothrombinase. So let's just say for right now that you got prothrombinase. So prothrombinase, which is also called 10A, and it's only represented as 10A on that diagram, okay? But I want you to know it by name. I don't have you know everything by name, just a few things. So 10A, so notice, here's 10A, okay? If you get 10A, you are going to get a clot, okay? We'll come back and figure out how you get 10A. So here's what 10A does, prothrombinase. Prothrombinase, which was never present before, which will convert this inactive protein called prothrombin to thrombin. Okay, before we go any further, please note that the naming isn't as hard as you think. Prothrombin literally means before thrombin. Prothrombin to thrombin. And what's the enzyme that converts it? Prothrombinase. Does that make sense? Okay. Now thrombin is going to convert another inactive protein called fibrinogen to fibrin. And fibrin is where we were trying to get, right? Fibrin, which is stabilized to form a clot. Does that make sense? Okay, which of these were always present? This one was always present, and this one was always present. Do they interact with one another if prothrombinase is not around? No, they don't. Right? That makes sense? Okay. Everybody good? So now we have a clot. Okay? Any questions over this part? Okay, so now let's go back and figure out how we get here. It's not as bad as it seems. There are two different pathways to get to prothrombinase. There is the extrinsic pathway, and there is the intrinsic pathway. I'm going to tell you a little bit about them, and you match them to which one's which. One of them is initiated by platelets from within the bloodstream. The other one is initiated by damaging the vessel wall or the tissue around it. Okay, which one of those matches with extrinsic pathway? Starting with platelets or starting with vessel or tissue damage? Tissue damage, that's extrinsic, right? The one that starts with platelets is intrinsic. Good? So that makes sense. Regardless, right, you're going to end up at the same place. The same place, extrinsic pathway, intrinsic pathway. If you get here, you're going to get a clot, so this is called the common pathway. And they both lead to it. Is that okay? Okay, so let's talk about the extrinsic pathway just a little bit more. Okay, so let's look at the extrinsic pathway here. Here's the extrinsic pathway. You do not have to know all of the intermediate steps, but what you do have to know is the extrinsic pathway starts with tissue damage, either the vessel or the tissue around it, which will ultimately cause you to release tissue factor 3, also called TF3. Damaged tissues release tissue factor 3 into the bloodstream, and tissue factor 3 will activate prothrombinase. That's how you get there, okay? So the deal is extrinsic pathway, tissue damage releases tissue factor 3. That make sense? And tissue factor 3 will ultimately get you to prothrombinase. Good? So vessel damage or tissue damage. Okay. Now, the intrinsic pathway is more complicated, but I'm not going to make you memorize it any more complicated. So look, the intrinsic pathway, if you get those platelets sticky and starting to call their friends, ultimately what they will do is release PF3. Instead of tissue factor, that one's platelet factor 3. So with this one, the platelets will release PF3. Tissue factor, platelet factor. Good? Okay? And PF3 will ultimately get you prothrombinase, yes? Okay? Okay, so, and I'm going to summarize this for you in just a second. So, when you get here, you are at the common pathway, and it doesn't matter how you got there, right? Now, what do you think, guys? If I damaged a vessel, which one do you think I would be doing? You'd be doing extrinsic, but then when the platelets started to notice the damaged vessel, you would start doing intrinsic. And if I wanted to plug the hole really quickly, isn't it good to do both at the same time? Yeah? Does that make sense? Okay. All right. And then we'll talk about pathology situations in just a second. Okay, you guys ready? Here's the summary. It'll make it way easier. Ready? Here's how you read this. The extrinsic pathway is activated by tissue damage, yes? Which releases tissue factor 3, which activates prothrombinase, right? That's where the common pathway starts, yes? And prothrombinase converts prothrombin to thrombin, and thrombin converts fibrinogen to fibrin, and fibrin is stabilized to form a clot. Good? Okay, let's read the other one. The intrinsic pathway, if those platelets get exposed to collagen, they'll start their platelet party, right? And those sticky platelets will release platelet factor 3, yes? And platelet factor 3 activates prothrombinase, when now you're at the common pathway again. And prothrombinase converts prothrombin to thrombin. Thrombin converts fibrinogen to fibrin. Fibrin is stabilized to form a clot. Does that make sense? Okay. So, I want to, before we move on and add a couple of other little factors that you have to know, I want to talk about my disorder and then also arterial or atherosclerosis. Okay, so I have atrial fibrillation, and one of the risks with atrial fibrillation is that because my atria do not contract dependably when I'm in atrial fibrillation, I run the risk of forming inappropriate clots, right? So what should happen is at the end of ventricular filling, right? Your atria should contract and push that last 20% of blood into the ventricles. But when I'm in atrial fibrillation, what happens is that blood just sits in my atria, right? Okay? Now, I want you to tell me which pathway is trying to kill me after that occurs. Which pathway? Which pathway? Extrinsic or intrinsic? It's the intrinsic pathway, isn't it? It's the intrinsic pathway. It's because the platelets are just sitting around, and if they just sit around, eventually they go, you know, I didn't see any collagen, but let's just go ahead and start a clot anyway, right? Because we're here, right? So what happens is the intrinsic pathway, if the platelets congregate for long enough, they will start a clot even without a damage. Does that make sense? Okay. Same thing happens with a person with arteriosclerosis. What's the problem with having really lumpy vessels because you've eaten loads of cholesterol and haven't exercised in your life? You might catch a platelet, and then it attracts two friends, and then it attracts two friends, and then it attracts two friends. And then it throws a clot and you have a heart attack or stroke, right? So it's the intrinsic pathway that can lead to inappropriate clotting. Does that make sense? Okay. That's why they want me to take aspirin every day. Okay. Is that good? Okay. Did you have a question?
Yeah. So, to store blood, they cool it, is that right?
They do, but I'm about to answer that question. Like, how does stored blood not clot? Yeah, hold on to it. I'm about to answer it, okay? All right, so that's a good question, though. Okay, there are at least 13 different factors involved in the coagulation phase. You are only responsible for the ones that were specifically pointed out in your notes. So do you have to know that prothrombinase is 10A? Yeah, you do, because I need you to be able to find it, right? And then you have to know these by name. You have to know tissue factor 3 and platelet factor 3. Do you have to know all of these? No, you don't. And if you learned them, would you remember them next semester anyway? No, it'd be like the damn forearm muscles. You'd be like, I know there's like 15 of them. I don't even remember what they're called, right? Some flexors and some extensors and some abductors and adductors. So you do have to memorize the big picture with a few relevant details. Notice that I'm only making you remember the details where they sort of change the story, right? This is an important part of the story. That one's a different story, and that one's a different story, but not all the intermediates, right? And so pass my class, because other physiology professors make you memorize the whole damn thing with all of the Roman numerals. Okay, so now, the other thing is that please note that, of course, calcium is required at multiple points. Which pathways? Look and tell me which pathways require calcium. Extrinsic, yes. Intrinsic, yes. And common. So if you do not have adequate extracellular calcium, you are screwed as far as blood clotting goes. Okay? That makes sense? Okay, so the other thing is, let me talk about vitamin K. Vitamin K is a lipid-soluble vitamin that is primarily made by your normal flora that is colonizing your large intestine. Okay? You've got normal flora colonizing your large intestine, makes vitamin K for you, and then you absorb it. Okay? Now, generally not a problem in an otherwise healthy person, but newborn babies have not colonized their intestine yet, right? And so they don't have adequate levels of vitamin K. What you need vitamin K for is to make this, okay? So if you don't have adequate vitamin K, can you clot adequately? No, because this is part of which pathway? This is part of the common pathway, right? So if you don't have adequate levels of vitamin K, you won't make prothrombin, and then even if you made 10A prothrombinase, you can't clot adequately. Does that make sense? So what did we do when we figured this out? We figured out that the whole birth and parturition process is potentially kind of violent, right? And you might have to poke a baby immediately afterward if you needed to figure out something else that was going on with them. And they are at relatively high risk of hemorrhage immediately after they're born because they don't have prothrombin. So what we do is we actually give them a vitamin K injection, right? And it protects them for about the first 8 days of their life until they have colonized their intestine and made their own prothrombin. The tragedy about this story is that mixed up with the poor education going on with the anti-vax movement is people misconstruing anything in a syringe as a vaccination And families have decided not to give their kids vitamin K injections, and we have had babies hemorrhage unnecessarily. Just because they think, oh, it's in a syringe, it's a vaccine. It's not a vaccine. It's a nutrient molecule that they can't actually make yet. And if they actually get injured within the first 8 days of their life, they're far more likely to hemorrhage if they don't have adequate levels of vitamin K. So this was an interesting, my OBGYN, so I had a high-risk pregnancy, so we got to talk about all kinds of this stuff, and I went back and talked to him just recently, and he's like, yeah, I have had 10 women over the course of the last 6 months say that they don't want vitamin K injections for their baby, and they actually had one fetal death just with his babies. Just because the baby just hemorrhaged because of lack of something that you could very easily add. It's really interesting. And she just insisted that it was actually toxins from the pharmaceutical industry. And I'm like, oh, a little knowledge is a really dangerous thing, apparently, deadly. Okay, so the next phase is clot retraction. This one's kind of interesting. So if we've got our clot, and we do, okay, apparently, I don't know who the hell discovered this. This was only within the last few years. This is a platelet right here, okay? Apparently, platelets, in addition to having clotting factors that they can release inside, also have actin and myosin. And what happens is the healing tissue will cause them to actually release different paracrine agents, which causes a little bit of cross-bridge cycling. And the platelet kind of shrinks, and since it's stuck in the spider web, it actually pulls the edges of the damaged tissue together. It's called the clot retraction phase. That's usually when, if you have a road rash, it will squeeze out that clear yellow fluid. Not pus. Pus is opaque. Right? The clear yellow fluid is serum. It'll squeeze it out during the clot retraction phase. Isn't that interesting? Actin and myosin, I don't know who discovered that. I think it's cool. Okay. And then the last phase is when you dissolve the clot. And this is called fibrinolysis. Because what's the clot made of? Fibrin. Yes, the clot is made of fibrin. So let's talk about fibrinolysis. This is the dissolving of a clot once the blood vessel is repaired. So it was made of fibrin, and what happens is the healing tissues, so if we have healing tissues, those release different paracrine agents than damaged tissues, right? And specifically what they will release is something called TPA, which is tissue plasminogen activator, and here's what TPA does. TPA interacts with an inactive plasma protein that's always around called plasminogen, which literally means the genesis or beginning of plasmin. TPA converts plasminogen to plasmin, and plasmin dissolves fibrin. Does that make sense? So as the tissue heals, right? It's like everything's cool here, there's no more hole. Then it releases something that says you can dissolve the clot. Which is kind of cool. So TPA has now been genetically engineered. We know how to make it in the lab. It's a really super duper effective, scarily effective clot buster. And it's really expensive. I don't know why it's expensive. It must be hard to actually get high levels of it produced in the lab. And if I had a stroke or was at a predisposition to a stroke and I had really good insurance, then they can give you TPA. And what it does is it's a really powerful clot buster, but it's really risky because if anything happens to you and you've got TPA in your system, you will hemorrhage. Go ahead. Uh-huh, yeah.
That's what they use to flush them out.
Oh, they do? They use TPA to flush out
Oh,
pipelines?
actually, the last time they injected it because the entire pickling goes all the way to your ventricle.
I would have thought they would use something cheaper, like heparin or something.
They say they use heparin.
Yeah, heparin is way cheaper. It's really easy to make.
Last time I was on there, like TPA.
Whoa,
Yeah.
that's fascinating that they would use TPA. For some reason, I don't know why, I think it seems like the last time I read it was the cost. They don't use TPA very often after heart attacks, but they usually use it after strokes. And I think it's probably just because strokes are so potentially massive with the damage that they can cause. So that's TPA, tissue plasminogen activator. This is not a medication that you will be sent home with, right? Right? This is a medication in which you are inpatient and they are actually taking care of you. So that's TPA or tissue plasminogen activator. Let me tell you a couple of other clotting considerations. Okay, so here's a question. What prevents, since a clot is kind of like this cascade, right? What prevents a clot from going and filling up the entire vessel? Well, there's a couple things. First off, just like the healthy tissue around releases stuff that tells the platelets to move away, right? The healthy tissue also releases anticoagulants, like antithrombin. There's a bunch of them that are just constantly being released by healthy tissues. So generally speaking, these active anticoagulants like antithrombin and heparin is constantly released by mast cells and basophils, and it just says, please don't clot here, please don't clot here, right? When I was pregnant, because of my heart problem, I had to, and estrogen, I don't know if you guys know this, estrogen increases your likelihood of clotting. So the combination of those two things and high levels of estrogen during pregnancy and my atrial fibrillation and my valve problem made it really likely that I would throw a clot during pregnancy. So I just shoot myself up with heparin 3 times a day the entire pregnancy. And what heparin does is heparin destroys thrombin, right? Heparin inactivates thrombin. So that's how that one works. So you are constantly releasing anticoagulants. And then in addition, you're sort of carrying away the activated clotting factors. So you only really clot in an area in which you accumulate activated clotting factors and don't have the anticoagulants, right? So it's kind of a numbers game, right? In a healthy person, you're constantly carrying away the activated ones and saying don't clot here, right? So it sort of localizes the clot, kind of like the platelet factor thing. Okay, so let's talk about a couple of different treatments with our last few minutes. So heparin is, of course, administered therapeutically, like during pregnancy. They also used to really commonly use heparin locks, right? Have you guys heard of a hep lock? They use saline now more often, but go ahead.
LDS hospital, they actually discovered that you don't have to use heparin in a saline lock.
Now they just use saline?
Yeah.
So they still use hep lock some places, but not all places. They used to, when you had an IV and you needed to not clot, they used heparin, and now they just use saline most of the time. They can use heparin during surgery, so they can use heparin. What about maintenance medications that they give as anticoagulants? So let's say I had hypertension and arteriosclerosis and I was predisposed to inappropriate clotting. They could give me maintenance medication, which they call blood thinners. They're not really blood thinners, they're anticoagulants. And what those, like Coumadin and warfarin, what those do is they actually tie up vitamin K. They're vitamin K antagonists. And so where do they affect this story? Where do they affect it? Yeah, they take out prothrombin, right? So you're basically like a newborn baby as far as your clotting factors. And you definitely do not want to take one of those. Also eat leafy green vegetables that makes you produce more vitamin K. Also take aspirin or ibuprofen, right? Because then you're going to bleed out like crazy if anything happens to you, right? So that's Coumadin and warfarin. That's how those like commonly referred to as blood thinners, but they're really not blood thinners. They're anticoagulants, right? And then the last one addresses your question. So how is it that I can give blood and have it sit in a bag, right, to be, for instance, donated to someone else without clotting? Because you would think it would just clot immediately, right? Okay. All they do is they actually give you something that attracts all the calcium out and sequesters the calcium. And remember, calcium was needed for intrinsic, extrinsic, and common pathways. So if you pull all the calcium out of the solution, you can't clot. So all it is EDTA, which is like a detergent that binds to the calcium, and it just keeps the blood from clotting. You also have to keep it cold so that it actually doesn't start to attract pathogens and that kind of stuff. And then they wiggle it before they put it back into you anyway. And then as soon as you shoot it back into somebody, their calcium just provides the calcium that is necessary, right? That's just the relationship there. Did they actually discover that at LDS hospital?
The heparin?
Yeah.
Yeah, they did like a study back in the 90s currently.
That's awesome. I didn't realize that happened here. We are also the place that we, the University of Utah was actually where they came up with the artificial heart. There's lots of cardiovascular research here. So we will do this last little part when I see you guys on Tuesday. And this will eventually be posted, but don't expect it until like Sunday or something. Okay?